Hello and welcome back to the channel, or indeed if this is your first time here, welcome to the channel. You're joining StudentSOS and we're really grateful to have you here. So this afternoon we're going to take a little look at cell mediated immune responses. This doesn't really follow in the order from DNA structure, but I've been working with a number of Year 12 students recently who've been really struggling with this particular aspect of immunity. I will do a video this week also of the non-specific immune response, but students struggle with that much less. So let's take a little look on the diagram at where we begin. So we have a pathogen and it invades the body and it breaches the non-specific barriers. We have a T helper cell and this recognises the pathogen as foreign and it carries foreign antigens as a result of that. This pathogen has foreign antigens, that's the orange triangles, on the surface of the pathogen. And it samples, can you see the T helper cell's receptor there? It's sampling some of this foreign antigen. So the T helper cell then travels to the lymph nodes. Now we have these, there he is, and we have these in our throat, we have them in under our armpits and in our groin. So in the lymph nodes, we have a couple of different types of cell. We have memory B cells and naive or immature B cells. So the memory B cells here have a little B and an M next to them, and then the naive or immature B cells just have the B attached to them. So the T helper cell then basically asks the B memory cells, have you seen this antigen? And it's very much like a wanted poster at a police station or online saying, have you seen this man? In this case, it's have you seen this antigen? B memory cells in this instance have not seen the antigen before. This is a new pathogen. So the memory B cells don't respond to the T helper cell. And so he takes one of the naive or immature B cells, doesn't matter which one. These guys have never been out before. They've never seen live action. So they haven't responded to an antigen before. So it doesn't matter which of them the T helper cell takes. So he picks one up and he takes them back to the site of infection. There they both are. And then they do two processes. Here we go, it's point 3. So upon return to the site of infection, the T helper cell and the B cell join receptors. And you can see that they're doing that there with the antigen between them. And this shares the information about the antigen structure with the B cell. And at this point, the immature B cell learns for the very first time how to produce antibody to this specific antigen only. And it will never be able to produce antibody to any other antigen after this, only this antigen. So at this point then, the B cell is performing something called clonal selection. This is called clonal selection. I'm going to tell you right now, some of the textbooks have the most appalling diagrams for this. The OCR AS level, if you're listening OCR, it really is a dreadful diagram on cell-mediated immunity. Please address that. But they really are very confusing and not at all clear. I think this makes it worse for students who are trying to learn this topic. So once clonal selection is complete, the B cell will now divide and make lots and lots of copies of itself in a process that you all have heard of is hopefully called clonal expansion. The B cell divides by mitosis, so all of the cells are identical. More on mitosis later on. And basically all of these B cells are able to produce the very same antibody because they're clones. You'll see that a couple of these cells become memory B cells, and these guys head back to the lymph nodes. There we go. And now they're stored as memory B cells. So if a T helper cell comes and samples the same antigen again, there will be a B memory cell that recognises it, and it will be able to return to the site of infection. We'll go back to that in a moment. In the meantime, I've got all of these, and they're plasma B cells, these blue ones, these purpley blue ones, they're plasma B cells. And these plasma B cells are able to produce antibody, and what they then do is they produce antibody, they move to the site of infection, produce antibody, and the antibody is able to attach to the antigen on the surface of the pathogen. Now, this causes neutralisation and subsequent destruction of the pathogen, so it can't cause any further damage to the body. I'm not going to talk about in this video exactly how neutralisation of pathogen occurs using an antigen-antibody interaction. I'm going to leave that for a separate video, but the words that you should be familiar with associated with this are agglutination and opsonization. So if you're reinfected by this same pathogen with the same antigens, what will happen is that the T helper cell samples again. It becomes an antigen presenting cell. There are other types of antigen presenting cell. This is just one, the T helper cell. But it's able to sample that antigen, go to the lymph nodes. They usually go to the nearest lymph nodes to the infection site, and this time be able to pick up a memory B cell. So when they return to the site of infection, there'll be no clonal selection needed this time, only some clonal expansion, and therefore the time that it takes to get to an infection and neutralise it is massively reduced, because now we do clonal expansion, we send some memory B cells back to the lymph nodes, and we take our plasma cells and they produce antibody against the antigen. And that hopefully means that you never actually have any symptomatic disease whatsoever if you are reinfected. This doesn't always happen, and there's a range of reasons for this. Very shortly, I'm going to do a very short video because it goes beyond the scope of the specification, but I suspect you'll find some questions related to it next summer in exams for A-level biology. We're going to take a little look at coronavirus and vaccination. Now, I'm very aware that vaccination has become hugely controversial, which is a shame because it's not controversial in the scientific community. So what I will do is tie together vaccination and the example being coronavirus in the next video so that we can take a little look at how vaccination actually works. In the meantime, please find us on Patreon. I'm going to do a short video in a moment about that. And find us on there. It's only £3 a month to join us. You get exclusive content. If you'd like specific one-to-one sessions with me, you're able to get a 10% discount. But please hop on over and indeed have a look for the video on Patreon, because we'll link Patreon in the information underneath the video. But in the meantime, please hit like if this was useful to you, and indeed subscribe for further notifications of new material. I am very much focused on Year 12 at the moment, because I'm aware that you have exams in the next 2 or so weeks in most cases. I'm also doing some more content on UCAT. So please, again, if you'd like to know more about these things, if you comment below, tell me what you're struggling with and we'll do a little video on it. In the meantime, thanks for joining me this afternoon. Have a fantastic day. Take good care. Bye-bye.